So now I want to introduce Georgina Stegmayer. So Georgina received the PhD degree from Politecnico di Torino in Italy in 2006. She is now a professor of artificial intelligence at UNL, University in Argentina. She's currently independent research scientist at CONICET and the head of the bioinformatics group at SIC, CONICET UNL. And her core research interests involve machine learning, data mining, and pattern recognition in bioinformatics. So thank you very much, Georgina, for being here today.
Hello, thank you. Thank you for the presentation and for the invitation for being here. I will present now. Okay, I hope you can see this. Is it okay?
Yes, now
yes.
We can see.
Okay, thank you. Well, yes, my name is Georgina and I come from Santa Fe, Argentina. I work at the Think Institute in city of Santa Fe. And the intention of my presentation is to show you all the work we have done in this last 5 or 10 to 10 years where my group has been growing up. I will present a lot of projects, several different projects. We are all machine learning people, so most of the presentation will be about machine learning with omics, different types of omics data. My group has a lot of people. There are some researchers in bioinformatics, and then we have PhD and postdoc students and some graduate students. And we have worked in many different kinds of projects which involve all of them machine learning. And so this is the page of my groups where we are all the team members and our publications and what we call web demo, because we think that one of the most important capability that bioinformatic tool must have is to be easy to use through a simple web server. So we try to make accessible all our models through what we call web demo, which is a simple page where you can input your data and try our algorithms. Well, I will tell you about our work in these last years in what we can say is a wide machine learning pipeline for different kinds of omics data. So we have experience and we have worked in the data preprocessing, normalization, and integration of different kinds of sources. Then we have also experience in which we call data mining into this biological data. We have worked in clustering and classification problems. And then at the last point, once we have found a new cluster or a new kind of relationships, we try to help in what we call the inference of new biological knowledge. And we are actually involved in 3 different kinds of projects. One is automatical functional protein annotation with machine learning. Then we have also worked with metabolic pathways, the inference of metabolic pathways and joint regulatory networks. So I will tell you all of these very quickly in this presentation, just to give you a glimpse of all our work. We have started our research group with this first project involved Solanum lycopersicum data in collaboration with Dr. Carrari in Argentina. And the challenge here was that there were a lot of different types of tomatoes with different properties, different genomics and metabolomics, sensory panels, a lot of measurements, different kinds of data was collected from this huge collection of different kinds of tomato. And the idea was to be able to integrate all this information for discovering new knowledge. So we started from these huge Excel files where there were the metabolites, the transcripts, sensory panels, a lot of different kinds and types of information, numerical and non-numerical. And so the problem here was how to normalize and integrate all of these in order to find some interesting relationships between them, so which could lead to new knowledge. for example, for improving biotechnologically the product. So we developed our first bioinformatics model, which was a self-organizing map, which is a kind of neural network, which receives a lot of data and puts this data into neurons in the model. And similar data goes into the same neuron. The assignation to the input data to the neurons is through for the minimum Euclidean distance, because all of these are numerical data which can be measured. So this way we were able to integrate into one model different kinds of and types of information which had to do with, for example, acids, metabolites, sugar, amino acid, shelf life, fruit ferments. So a lot of different, very different kinds of aspects of the tomato. And then with this model, we develop a web server, which is free to use. And you can put all your measurements, different kind of measurements, and they will be integrated into this machine learning model, which is Omicom. And when there are neurons that get different kinds or different types of information, for example, here, metabolites and transcripts, these neurons are painted in black, and if there is only transcripts, the neurons are painted in red. So this way, graphically, you can very easily see where is the integration of different kinds of data, and you can go into there and see the detail of the measurements, the values of the data that has been clustered there. Then, as a continuation of this project, there were another collection of tomatoes coming from different regions in Argentina, and the idea was trying to discover the particular properties that they have in order to resend this line of tomatoes into Argentina. So there were a lot of quantitative and qualitative measurements that were obtained on these materials, volatile metabolites expression, gene expression, and tasting panels. So all of this information was used differently, and so different cluster algorithms were used, and then an ensemble cluster model was proposed in order to fuse all this information into one solution. So what we have was different kind of types of tomatoes, different characteristics, sizes, and colors. As you can see, there are some features that were not numerical, that were using categories, or only 2 or 3 values with labels. So we proposed a model which was able to cluster every different category separately, and then put it all together into one single model. So this way you can integrate not only numerical data, but also categorical data and data with different kinds of labels without doing any pre-processing, without the need of any normalization. So this was a very complex model, but it was able to use any kind of data without almost any pre-processing for the biologists. Then, once we have found some clusters with data integrated, different kind of data integrated, the next step was how can we extract knowledge or infer knowledge from these relationships? For example, all the metabolites that are inside one cluster, are they maybe forming part of the same metabolic pathway? So we developed one line of research which involved the development of automatic way of finding the pathway that was connecting all these metabolites in one cluster. So one researcher of my group started to work in evolutionary algorithms, which were able to represent with a chromosome, artificial chromosome, and then do evolutionary mechanism in order to find a regulation path which involved all these metabolites, and these were found automatically with these algorithms. Another research line involved, well, we have a lot of measurements of genes over time. We have here at the left top, there are several genes that were measured on time or in different experimental conditions. We want to find whether there is a hidden regulatory network among these genes, which is hidden in this large amount of measurements. So in this work, another researcher from my lab work in the automatic finding or inference of the trend regulatory network, which was suggested in this data with genes and time measurements. And the solution he proposed involved multi-line perceptron models and extreme learning machines. So this is supervised machine learning neural networks, which use the input and output as input and output the sorry, the time signal of the genes. And so there are a lot of neural networks that are built trying to see whether one signal of a gene is the better predictor for the next signal. So when the model has the shortest error, this is marked as a possible connection or regulation between those genes that were analyzed. So this way, trying all genes against all genes and with some rules, there is a way of finding a matrix which can represent and gives light to the hidden regulatory network that exists between these analyzed genes. Then, well, more recently, we started to work in the problem of inference of the gene ontology terms. As most of you know, you have genes and proteins which are annotated functionally with gene ontology terms in all the three subontologies, but many times happens that there is a gene which is actually has a present expression. It can be measured in an experiment, but it has no annotation. It's new, and nothing is known about this new gene or protein. So the aim here in the project is to be able to assign automatically with machine learning some gene ontology terms to this new or newly discovered protein. So in the panel of A and C, you can see that for all genes in an experiment, there is an expression matrix, and the panel B shows a distance expression matrix that can be calculated. But in panel C, you see that some of these genes have gene ontology annotations, which we call semantic data, but many of them don't have. So the matrix in the panel D, called semantic distance matrix, cannot be fully calculated because there are some genes where you cannot calculate the distance in terms of gene ontology terms because there are no terms. So we have one of the researchers of my group and a team, we have developed an algorithm which uses non-negative matrix factorization, which is a mathematical tool which can complete a matrix using the information from another matrix. So there's a sketch of the algorithm that basically it allows to reconstruct mathematically the semantic distance between the genes which have no annotations, and then from those from this semantic distance which is reconstructed, it is possible to with the Bayesian method assign actually gene ontology levels to these genes which were previously unknown. In this same line of work, we have another researcher which is here now and will be presented in the next session, Gabriela, and he's actually working with protein function annotation with deep learning. So from proteins and from genes, there are a lot of different kinds and sources of information, not only sequence, but also the occurrence in racton pathways, the occurrence of the annotation in other subontologies of the gene ontology, and then there is a way of calculate the distance in the protein sequence distance in addition. So all of this information and many other more can be used as inputs to a deep neural network, which has many layers. And it is possible to use this model to infer different levels of the gene ontology terms, the upper levels and the more specific levels. So Gabriela has designed this model, which was able to realize this task very well. We have here the technical details of the model, which has some dropout probability, but sites, etc. Lots of combination of parameters were looked for in different data sets, We have found a model which integrating all this kind of different information from the available in the different databases from proteins. It is capable of predicting the gene ontology terms with high precision for these proteins that were previously completely unknown. Well, and in the last part of my talk, I will talk about our more recent project, which involved part of my team last year, because we were working also in parallel with other projects. We were working a lot in pre-microRNA prediction, which means that there is a database of pre-microRNAs, which are a small sequence of nucleotides, and there are lots of them which are known in animals and plants and virus. And the intention is when there is a newly sequenced genome, to be able to find if inside of in this new genome there are some pre-microRNAs which can have some important function, because they had to do with regulation of cells and infection and diseases. So there is a lot of interest in finding them, not only with measurements, but with the computational methods, looking only at the genome. So we have worked a lot in tools and in different models which are able to take a genome, cut it, and do the folding of the sequence to see if they have this pre-microRNA typical hairpin shape. Then there are a lot of feature extraction algorithms which transform this into numbers that go into a machine learning model, which is able to predict, if you cut a genome into pieces, if this piece will probably be a new microRNA or not. So we, as I said before, pre-microRNAs have features which are widely known and can be extracted in order to represent every piece of a genome that one you are analyzing. Then when we have developed many tools for feature extraction, up to date, all of the features that were published up to this year were made in a tool that is available. And then we have also developed how to extract pieces of a genome and check if at least they can be hairpins. And so you can filter all the parts of the genome which will not be hairpins, and so there will not be good candidates to Premicrornas. And more recently, one of my PhD students has developed a new feature, because there are many features available in literature, but he has proposed in his thesis a new feature which is able to further improve the classification of machine learning classifiers, because it looks into the edit distance which is necessary from one sequence to get to another one. This can be counted, and this can be used as a feature between several sequences, and it has been tested that it has improved a lot the classification of this kind of sequences. His more recent work involves the use of transformers, which is a very, very new kind of deep learning model, which needs no pre-processing. So there's no need to cut the genome into sequences, look for features, and then put it into a classifier. This kind of deep learning model look the raw sequences, the raw genome-wide data. It's kind of slide a window, and looking at these species without any feature extraction, they automatically extract features internally in the deep model, and then they are able to classify each piece that is looking as probably Premicrorna or not. We have in this line of work, we have developed many other tools, many other models, which were based on self-organizing maps for the discovery of new pre-microRNAs in genomes that were recently found. Particular, one of our best models was based on was called deep ensemble elastic self-organizing map, because at the beginning, at the first layer, there is an ensemble of self-organizing maps that then filter data to different layers of self-organizing map, which they automatically determine the size of the map according to the data they have to cluster into the neurons. And we have proved this model with the hen and white data, and it was quite good. And so finally, my last slide. During the pandemic last year and at the beginning of the pandemic, we saw that we had all these tools and models developed for the analysis and the prediction of pre-microRNA in any genome. And we have used it a lot in plants and in animals. So we say, well, as a group, as a research group, how can we contribute to this pandemic that was occurring? So we thought, well, maybe it can be useful for some diagnosis or a vaccine, or I don't know. As a bioinformaticians, we can contribute using applying all the models we have already developed for animals, trying to see if there are some pre-microRNAs in the SARS-CoV-2 virus. So we developed this pipeline where we took the novel virus, we cut it into pieces, and we extracted features to fit some machine learning models which need features, and then we use it in a novel deep learning model that we were developing at the time, which didn't use features. It was able of taking directly the genome without extracting features. So we say, well, what about if we combine all our models and experience in this subject, and try to see if combining several machine learning classifiers, we found some pieces of this virus which can be pre-microRNAs and can have important, interesting functionality, which can be used for diagnosis or treatments or whatever. So we put together this group of these people, some of my group, some other biologists, collaborators. So we did this work. We train looking at the already deposited microRNAs that are available for virus in MIRBase, which is the database of well-known microRNAs. We train all our 3 machine learning models. We use a one-class support vector machine, which is always used in this field. We train the DSOM, and we train the MIRDNN, which is a convolutional neural network that takes the raw pieces of the raw sequences and the matching or unmatch of the nucleotides when they form a hairpin, and extract a feature which is the folding energy. So I'm using all these. There are several layers of convolutional layers with identity blocks, which are known as ResNet. So we put together these 3 models. So we fit the models with the non-virus microRNAs deposited in MIRBase as the positive class. So we are trying to look for something similar to this into the novel SARS-CoV-2 virus genome. So each model independently look for the best candidates to be novel pre-microRNA. And since it was, let's say, critical task, we selected only the best candidates that were indicated by all three methods combined. So each method had a ranking of candidates, but we selected those that were in the intersection of the 3 methods. Well, our research could lead up to this point in May 2020, because there were not no experimental validation. There were no measurements of infected cells. But in May 2020 appeared a free database which we could use to see if our predictions could be validated with experimental data. And effectively, from all the candidates we have found, we could check and validate that some of them have already had reads of expression after 12 and 24 hours of infection with the virus. So these are the detail of one of the candidates to Premicrorna in the virus we have found. And so these are the experimental measurements that can be found with the tissue exposed to the virus for 12 and 24 hours later. And the region of the Premicrorna that our machine learning models have identified as the region where the mature microRNA can be, it was actually corresponding to the part of the genome where the highest expression was found. So we were able to validate experimentally our computational prediction. And this is another example where we not only saw that the predictions some of them were validated with experimental measurements, but we wanted to see which part of these sequences were those that the deep learning model was looking at, because the deep learning model doesn't need features, so it just look at the raw data, then extracts internally the features. And there is a way of measuring the attention that the model puts to some parts of the sequences that it's analyzing. So we did that, and we saw that the parts that the model was taking more attention to were those that actually then had reads. So those parts that were actually important for the microRNA to express. So, well, we were very happy with this discovery, and we could publish it. We hope that it is helpful for some treatments or vaccine, or I don't know. My machine learning knowledge arrives here, and we put a small stone so that somebody can build on top of it. And that's all. Thank you.
Thank you, Georgina. Unfortunately, we almost run out of time. There is a small question in the chat from people working at INTA, from Sofia Eileen. Wonder if she can use the same neural network that you use for tomato in sunflower data.
Yes, sure. Yes, of course. Yes, because yes, it's a machine learning model, and the important thing is to put the correct data into it. But yes, there's no problem. I think it could be directly translated to another domain. Maybe there is some preprocessing of the data that has to be done, but I'm sure it's possible.
Okay, thank you, Georgina.